Today we're on the cusp of something truly remarkable. We're about to delve into a story of scientific ingenuity, unwavering determination, and the potential to rewrite the future for millions. It's a breakthrough in the fight against breast cancer, a relentless adversary that has cast a long shadow over countless lives. This is a disease that affects millions worldwide, transcending borders and impacting families in every corner of the globe. The statistics are staggering, but behind each number is a personal story of courage, resilience, and the enduring hope for a brighter tomorrow. For years, scientists and doctors, the unsung heroes of our time, have tirelessly pushed for better treatments, dedicating their lives to unraveling the complexities of this disease. They've explored every avenue, tested countless hypotheses, and faced numerous setbacks with unwavering resolve. And now, after years of relentless pursuit, we stand on the brink of a game-changing discovery, a moment that could redefine the landscape of breast cancer treatment. This isn't just another incremental step forward. It's not merely a refinement of existing therapies. It's a fundamental shift in how we understand and combat breast cancer, a paradigm shift that challenges conventional wisdom and opens up entirely new possibilities for treatment and prevention. The real danger of breast cancer, the aspect that makes it so formidable, is metastasis. This is when cancer spreads to other parts of the body, transforming from a localized threat into a systemic challenge that is far more difficult to manage. It's the process that often determines the long-term prognosis for patients. Researchers, in a moment of profound insight, have uncovered a hidden mechanism, a previously unknown pathway that allows cancer to spread, to colonize new territories within the body, and crucially, they've discovered a way to potentially block it, to disrupt this deadly process and prevent cancer from metastasizing. This discovery could change everything for patients and families, offering a new lease on life and the hope of a future free from the fear of recurrence. Today, we'll break down the science, demystifying the complex mechanisms at play and explaining the intricacies of this groundbreaking research. We'll explain what this means for treatment, exploring how this discovery could translate into new therapies and improved outcomes for patients. And we'll share why there's real hope on the horizon, why this breakthrough represents a turning point in the fight against breast cancer. Let's dive into this story of resilience, innovation, and optimism, a testament to the power of human ingenuity and the unwavering spirit of those who refuse to give up. This is a moment of hope for everyone touched by breast cancer, a beacon of light in the darkness, and a reminder that even in the face of the most daunting challenges, progress is possible. To understand this breakthrough, we need to look at metastasis, the process that makes breast cancer deadly. Cancer cells break away from the original tumor and travel to organs like the lungs or brain. Most cancer deaths are caused by this spread, not the initial tumor. These cells are like fugitives, evading the immune system and manipulating their environment to survive. They corrupt immune cells, turning them into unwitting allies. The journey is perilous, and only a few cells succeed in forming new tumors. Scientists suspected cancer was hijacking the body's own processes, especially metabolism, to aid its spread. This led to a crucial question: what in the body's environment is cancer exploiting? The answer would reveal a secret weapon in cancer's arsenal. The heart of this groundbreaking discovery lies within a seemingly ordinary molecule, uracil. It's a fundamental building block of RNA, a close cousin to DNA, and plays a crucial role in various cellular processes. But its involvement in cancer metastasis was previously unknown, making this finding particularly significant. Uracil is a molecule usually found inside cells, quietly doing its job, participating in essential functions like protein synthesis and enzymatic reactions. It's a ubiquitous component of the cellular machinery, constantly at work to keep the cell functioning smoothly. Researchers, driven by the need to understand the complexities of cancer spread, found that, near breast tumors, something unexpected was happening with this common molecule. They noticed an unusually high concentration of uracil in the vicinity of the cancerous tissue. Further investigation revealed that certain immune cells, specifically macrophages, were being tricked into releasing uracil outside the cell, a process they wouldn't normally undertake. This release was highly localized, concentrated around the tumor microenvironment. The insidious nature of cancer became apparent as scientists discovered that cancer cells reprogrammed macrophages, immune cells meant to fight cancer, turning them into accomplices in their metastatic journey. These reprogrammed macrophages were now acting against their intended purpose. The cancer cells manipulated these macrophages to overproduce and eject uracil, flooding the surrounding area with this signaling molecule. This overproduction was a key step in the cancer's strategy to spread. This uracil acted as a beacon, a chemical signal broadcasting the location of the tumor and paving the way for its dissemination. It was an unexpected and crucial piece of the metastatic puzzle. The uracil beacon was effectively guiding cancer cells and helping them invade new tissues, establishing secondary tumors in distant locations. This process, known as metastasis, is the primary cause of cancer-related deaths. Cancer cells, it turns out, have special receptors to detect this uracil trail, specialized proteins on their surface that bind to uracil with high affinity. This allows them to sense the direction and concentration of the signal. By detecting the uracil gradient, cancer cells are using it to migrate and spread, following the chemical trail to new sites in the body where they can establish new colonies. This directed migration is a critical step in the metastatic cascade. It's a devious partnership, a carefully orchestrated manipulation of the body's own defense mechanisms. Cancer hijacks the immune system, turning it from a protector into an unwitting accomplice in its own spread. And in this manipulation, cancer is using uracil as a superhighway for metastasis, a clear and direct route to invade new tissues and establish secondary tumors. This changes our entire view of how cancer spreads, offering a new perspective on the complex mechanisms that drive metastasis. By identifying uracil as the key accomplice, the molecular beacon guiding cancer cells, scientists have found a specific target to disrupt metastasis, a new avenue for developing therapies that can prevent cancer from spreading. Breaking this link, disrupting the uracil signaling pathway, could stop cancer in its tracks, offering a new hope for patients facing metastatic disease. Armed with this crucial knowledge about uracil's role in cancer metastasis, researchers began to explore a bold new question. What if we could somehow erase the uracil trail, effectively making these corrupted immune cells invisible to the cancer's spreading signals? Could we disrupt the communication that allows cancer to colonize new areas of the body? To test this hypothesis, they meticulously targeted the enzyme UPP1. This enzyme is specifically responsible for the production of uracil within these corrupted immune cells. The idea was that by inhibiting UPP1, they could reduce or eliminate the uracil being produced, thereby disrupting the metastatic process. To rigorously evaluate the potential of this approach, the researchers designed a series of mouse experiments. One carefully controlled group of mice received a UPP1 inhibitor, a compound designed to block the enzyme's activity. This was the treatment group, the focus of intense observation. A second, equally important group served as the control. This group did not receive the UPP1 inhibitor, allowing the researchers to observe the natural progression of the cancer without intervention. This comparison was crucial for determining the true impact of the UPP1 inhibitor. As the experiments progressed and the data began to pour in, the results were nothing short of stunning. The scientists were amazed by the clear differences emerging between the two groups. In the control group, the mice without the inhibitor, the cancer spread as expected. The malignant cells aggressively metastasized, colonizing distant organs and establishing secondary tumors, following the typical pattern of the disease. However, in the mice treated with the UPP1 inhibitor, the story was dramatically different. Metastasis was significantly reduced, in some cases almost entirely stopped. The spread of cancer cells to other parts of the body was effectively curtailed. While the primary tumor remained in place, the cancer's ability to travel and establish new colonies in other organs was severely hampered. The inhibitor acted as a barrier, preventing the cancer from gaining a foothold elsewhere. Even more encouragingly, blocking UPP1 had a positive effect on the immune system. It restored the immune cell's inherent ability to recognize and actively fight the cancer cells, turning the body's own defenses back against the disease. This innovative treatment not only halted the relentless spread of the cancer, but also reactivated and strengthened the body's natural defenses, creating a powerful synergistic effect. This dual action, targeting both metastasis and immune function, offers a compelling new strategy in the fight against breast cancer. The approach is simple yet profound. Stop metastasis in its tracks and simultaneously empower the immune system to eliminate remaining cancer cells. This represents a profound shift in our understanding of how we might effectively treat breast cancer. moving beyond simply targeting the primary tumor. These encouraging lab results offer a significant beacon of hope, illuminating the path towards the development of more effective and targeted therapies in the future. The crucial next step is translating this remarkable success from the laboratory bench to human clinical trials, bringing the promise of this treatment closer to patients in need. This breakthrough opens the door to a new class of drugs targeting metastasis directly. Imagine a pill that blocks UPP1, used alongside chemotherapy or radiation, to keep cancer from spreading. Instead of just attacking tumors, we could build a firewall around them. This approach could help early-stage patients keep cancer localized and give advanced patients a way to manage the disease long-term. UPP1 inhibitors are highly specific, potentially causing fewer side effects than traditional chemotherapy. It's about outsmarting cancer, not just overpowering it. While these findings are early and based on animal studies, the path forward is clear. Pharmaceutical companies can now develop and refine UPP1 inhibitors for human trials. This discovery gives real hope for stopping metastasis before it starts. Beyond treatment, this discovery could revolutionize early detection. Uracil outside cells is a unique cancer signature, a potential biomarker. A simple blood test could measure uracil levels, offering early warning even before tumors are visible on scans. For diagnosed patients, tracking uracil could predict metastasis risk and guide treatment choices. This could personalize therapy, ensuring each patient gets the right care. It's a step toward truly individualized medicine. By seeing cancer's next move, doctors can intervene earlier and more effectively. Early detection and prediction could save countless lives. While science advances, awareness remains our best defense. Breast cancer is the most common cancer in women, but men can get it too. Early detection is key. Know the signs: a new lump, swelling, skin dimpling, nipple changes, or unusual discharge. Not all lumps are cancer, but any change should be checked by a doctor. Don't ignore symptoms or feel embarrassed. Your health comes first. Practice breast self-awareness. Know how your breasts normally look and feel. This isn't about fear, but empowerment. Early detection saves lives, and it starts with you. Be proactive, informed, and attentive to your body. Encourage loved ones to do the same. Knowledge is power in the fight against breast cancer. Stay vigilant and advocate for your health. Regular self-exams are a simple, powerful way to catch changes early. For women, do them monthly after your period or on a set day. Look in the mirror for changes in size, shape, or skin. Check for lumps or unusual discharge. Feel your breasts lying down using a pattern to cover the whole area. Men should check too. Look for lumps under the nipple or chest changes. Most changes aren't cancer, but always get them checked. Self-exams take minutes but can be life-saving. Consistency is key. Make it a monthly habit. If you notice something new, don't panic but see a doctor. Early action makes all the difference. Take charge of your health. Your future self will thank you. This discovery is more than science. It's a symbol of hope and progress. We're moving closer to a future where breast cancer is less deadly and more manageable. But hope needs action. Support research, fund innovation, and advocate for access to care. Share this knowledge, encourage self-exams, and empower those around you. Let's replace fear with strength and stigma with support. Be proactive about your health and help others do the same. Every step forward honors those who face this disease. Together we can build a healthier future. Stay hopeful,